Hey everyone, I'm Pierce and this is my dear friend Peaches.
Hello guys, are you wondering what this video is about? Well, wonder no more. We are going to talk about a very interesting theme from the field of genetic engineering.
Yes, we are going to explore the procedure for increasing the copy number of your gene of interest. Okay then, before beginning our journey, let me take you through our fascinating checkpoints. First off, I'll give you a gist of how this tour works. And then when we reach to the second checkpoint, you'll get to see how amazing PGMT is. I can rest assure you that it is one of the best destinations.
Oh yeah, Pierce, that's an awesome checkpoint as you mentioned. Can't wait to get there.
Come on, Peaches, hold your horses and let the viewers have an idea about our trip.
Oh, sorry Pierce, you go ahead.
Our third checkpoint is the gene of interest. Hey Peaches, would you like to take it from here? I can see how eager you are already.
Thanks Pierce, I would love to. So, as you see, we would call it as a GOI. Though it's a small region to visit, without visiting it, your tour won't be complete. Then next, we would see how vector and GOI come together to lead us to our desired destination. That is what we would see in cloning.
Most importantly, don't you want to know if you are on the right track or not? So we'll take some breast and media screening and ensure it.
Hey, okay, and there you go, we will be at our destination.
What is the vector? Is there any special property needed to clone? How will they both help us reach our destination?
Are you gonna teleport over there? Then how will cloning and GOI come together?
Peaches, I suppose you meant how are we going to get to our destination?
Yes, Pierce, all these questions were confusing me when we were new to this, right? And the questions were never ending. Actually, what's a bioreactor? Do we need to select one? And how we know that we have the insert in our product?
Really, Peaches? These questions do sound overwhelming. Come on, let's not confuse people anymore and get into the video.
The very first step in our journey is the establishment of the vector. Am I right, Pierce?
Yes, after considering many factors, we have decided that our vector of choice is PGMT plasmid.
The PGMT vector is a high-efficiency TA cloning vector which contains a multiple cloning sites. Actually, this is a 3 kb in size and contains the ampicillin-resistant gene for selection.
Whoa, wait a minute. Before moving on to the next point, I think you should explain what TA cloning is.
Yes, of course. This method takes the advantages of A overhang added by the PCR enzyme like a tag DNA polymerase and T vector or linearized plasmid that has been treated to add a 3' T overhang to match the A overhang of the insert. That insert is directly ligated to the T-tailed plasmid vector with a T4 DNA ligase.
That's quite an explanation, Peaches.
Yeah, thank you, Pierce. And as I was saying, the PGMT vector system or convenient system to clone PCR product generated by certain thermostable polymerase. Let's take a look at the structure of PGMT vector.
In the PGMT vector, T7 and sp6 RNA polymerase promoters flank a multiple cloning region within the alpha peptide coding region for beta galactosidase. This region plays an important role in the selection of recombinants, a process which we'll be discussing in detail in a while.
And the vector also have a F1 origin of replication which aids the preparation of single-stranded DNA.
The multiple cloning site is flanked by recognition sites for the restriction enzyme BSTZ1, allowing release of the insert by a single enzyme digestion.
That's cool. Now, we are familiar with our vector. Am I right? Okay, let's move on to the protagonist of our story, the gene of interest, the transforming growth factor beta 1.
TGFB1 is the best characterized isoform of the TGFB superfamily. It encodes a secreted ligand of TGFB superfamily. Ligands of this family bind to various TGFB receptors, leading to recruitment and activation of certain transcription factors that regulates gene expression. Thus, the encoded protein regulates cell proliferation, differentiation, and growth. It can also modulate the expression and activation of other growth factors, including interferon gamma and TNF alpha.
And the protein encoded by this gene is a multifunctional protein that regulates the growth and the differentiation of various cell types and involved in a various processes such as a normal development and immune function.
The protein is found throughout the body, but is particularly abundant in tissues that make up the skeleton, where they regulate the formation and growth of bone and cartilage.
TGF beta 1 is also involved in the formation of blood vessels, development of muscle tissue and body fat, wound healing, and inflammatory processes in the immune system, and the prevention of tumor growth.
Here is a visual depiction of the TGF beta 1 protein. Hey Peaches, did you know that TGF-B1 is linked with obesity?
Oh wow, is it?
Yes, I would love to share an interesting fact about the role they play in obesity.
Yes, please. I would love to learn about it.
The release of TGF-beta 1 by human adipose tissue affects obesity in human beings.
Oh, is that so?
Yes, it was also examined whether its release is due to the adipocytes or the non-fat cells in adipose tissue. It was also observed that there is a significant correlation between the BMI of fat donors and release of TGF-beta 1 by the adipose tissue.
Wait. I also have a piece of information about it. The release of TGF-beta 1 over 48 hours by the adipose tissue was significantly enhanced both in the presence of inhibitor TNF alpha and IL-1 beta. How amazing is that?
I know, right? Also, the release of TGF-beta 1 was primarily by the non-fat cells of human adipose tissue.
Interesting.
Once the vector and gene of interest are established, we shift our focus towards the cloning process. The another method of cloning available is PCR cloning, otherwise known as TA cloning. In this process, the DNA fragment of interest and even the vector are amplified by the polymerase chain reaction PCR and ligated together without the use of restriction enzymes.
As far as the PGM-T vector is concerned, two modes of cloning are applicable: the traditional cloning system, in which the restriction endonucleases are used to generate the DNA fragment with a specific complementary end sequence that can be joined together with a DNA ligase prior to the transformation. Actually, the traditional cloning is the low-cost method which easily supports the directional cloning. However, the range of the vector which can be used is limited.
Despite the higher cost and the difficulties faced during directional cloning, PCR is preferred in some cases due to the large range of vectors compatible with the process as well as the lower runtime compared to traditional cloning.
Pierce, have you ever wondered what makes TA cloning special?
Of course, Peaches, TA cloning doesn't require any restriction enzymes nor does it involve any complicated steps. It simply makes use of the base pairing between adenine and thymine for cloning.
Absolutely correct, Pierce. This makes me wonder, how is it even possible? Wow!
Have you heard of Taq polymerase, the one which helps to generate PCR products?
Oh yes, I have. To be more precise, a Taq polymerase adds a overhang to the 3' end of the products.
Yes, and that is all you need. Your linearized vector that has a T overhang can be ligated to it easily.
Sounds interesting. Whoo! I think this picture itself speaks more.
True, Peaches, it does. See how A overhang and T overhang are helpful in this process.
Voila, you have cloned the plasmid ready at the cheap cost and the simplest way.
Well, Peaches, you know, right, that I'm kind of traditional when it comes to cloning. Yeah, I do love the new techniques out there, but the traditional way always satisfies me.
Don't worry, Pierce, you can achieve the same result via the traditional cloning. It's just that you got to perform a few additional steps.
Oh yes, that is not a problem for me. Let me explain it. So the restriction enzyme is the BSTZ1 here.
So, Pierce, what is the reason for selecting BSTZ1?
Okay, Peaches, the PGMT vector has multiple cloning sites, right?
Yes, right.
Yes. So the restriction site of BSTZ1 is one among the multiple cloning site of the PGMT vector. So here is the depiction of the restriction enzyme.
Okay. With my knowledge, I think the first step is a vector preparation. Am I right, Pierce?
Yes, Peaches, you're absolutely right. The first step is the vector preparation. So here we have the extracted plasmid and we will digest the vector with the help of restriction endonuclease BSTZ1. Secondly, we dephosphorylate the ends of the cut plasmid to ensure they do not stick back. This dephosphorylated plasmid goes for purification.
Ho, this is interesting, Pierce.
Haha, Peaches, we are here with the plasmid ready in our hands.
Hey, super, we are ready. So, Pierce, as we are ready with the plasmid, right, we will now start preparing the insert GOI.
Yes, please.
First of all, we will cut our gene of interest with BSTZ1. Since both the vectors and GOI are cut with the BSTZ1, both will have a blunt end.
Now to ligate the blunt-ended vector and DNA, we will be using appropriate linkers, right?
Yes, Pierce, perfect. Hey Pierce, do you know how the ligation process works?
Yes, Peaches. We will be using T4DNA ligase to ligate them, right?
Absolutely. The T4DNA ligase connect the DNA between the 3-OH group and the phosphate group.
Hey, Peaches, could you make it a little more clear?
Yeah, sure. Wait, here is a picture for you. Hope you get a better understanding now.
Yes, Peaches. This makes it clear.
Once the gene of interest is inserted into the plasmid contains the gene of interest that must be transformed into the host cell for multiplication.
In our case, we will be transforming the plasmid into an E. coli cell using electroporation.
An electroporation is a physical transfixion method that uses an electrical pulse to create a temporary pores in a cell membrane. Through the pores, which substances like nucleic acid can pass into the cell.
E. coli cells are incubated with the plasmids in an electroporator, where component cells and plasmids are exposed to a brief pulse of high voltage electric field. This treatment is believed to induce transient pores in cell membrane, which permit the entry of plasmids into the cells.
Hey, we have successfully transformed some of the cells in the population, Pierce.
Yes, but I wish we had a technique to efficiently transform the entire population.
Me too, Pierce. We will have to wait for such a technique to be developed. Until then, we can screen the population for recombinant.
Here, we will be using a technique called blue-white screening to differentiate the recombinants from the non-recombinants. The presence of lactose in the surrounding environment triggers the lactase operon in E. coli. The operon activity results in the production of beta-galactosidase enzyme that metabolizes lactose. The host E. coli strains used are competent cells and also contain lacZ del M15 deletion mutation. When the plasmid vector is taken up by such cells, due to the alpha complementation process, a functional beta-galactosidase enzyme is produced.
Do you guys remember the structure of PGMT vector? We had seen that the two RNA polymerase promoters T7 and sp6 flank a multiple cloning region within the alpha peptide coding region for beta galactosidase. This alpha peptide coding region is otherwise called as a LACZ sequence.
This multiple cloning site is nicked for insertion of the gene of interest when a plasmid vector containing foreign DNA is taken up by the host E. coli. The alpha complementation does not occur. Therefore, a functional beta-galactosidase enzyme is not produced.
If the foreign gene of interest is not inserted into the vector, or if it is inserted at the location other than the MCS, the LACZ gene in the plasmid vector complement the lag Z deletion mutation in the E. coli cell, producing the functional enzyme.
For screening the clones containing recombinant DNA, a chromogenic substrate known as X-gal is added to the agar plate. If beta-galactosidase is produced, X-gal is hydrolyzed to form 5-bromo-4-chloroindoxyl, which spontaneously dimerizes to produce an insoluble blue pigment called 5-5'-dibromo-4,4'-dichloroindigo.
Therefore, the colonies found by the non-recombinant cells appear blue in color, while the recombinant ones appear white. The desired recombinant colonies can be easily picked and cultured.
Here is a pictorial representation of the blue-white screening process.
We can easily observe that the plasmid in which the gene of interest is inserted into the lag Z sequence produce the white colonies and the others produce the blue colonies, thereby enabling us to differentiate between the recombinant and the non-recombinant.
This is an actual plate on which blue-white screening was done. The blue and white colony are visible.
Wow, it looks beautiful.
Now we have reached the final step of our process.
Woohoo, that's great. In this step, we will increase the copy number of our cells in order to obtain a substantial amount of our gene of interest.
The transformed E. coli cells acts as a bioreactors in which the plasmids replicate.
Apart from that, the E. coli cells also proliferate to produce more cells, which contain a plasmid bearing the gene of interest.
Thus, more and more copies of the gene of interest are produced.
And finally, we have successfully increased the copy number of our GOI pairs. Hey, woohoo!
Yay, what a journey it has been. Peaches, do you remember Sam Keene's immortal words, genes are like the story and DNA is the language that the story is written in?
Yes, of course.
Well, today we wrote quite a story of our own.
Yes, and we hope that we have inspired our viewers to go out there and to explore the wonderful world of genetic engineering and to write their stories of their own. So happy and thank you everyone for co-occur with us on this wonderful journey. Thank you and love you all.
I hope that you all have learned something valuable today. If you enjoyed the video, be sure to like, share, comment and subscribe. This is Pears and Peaches signing off. Bye.